So we talked about classification of media based on the physical state. Thank you for reminding. Based on the physical state and based on the chemical composition. So the next thing to focus on is the function of the medium. Okay. The TSA Agar, which I'm going to use a lot as an example, is the general purpose. Is anything that's of the general purpose, it's okay for a lot of things, but it's not perfect for pretty much all of these things. Does that make sense? It works good to grow microbes, okay? All kinds of microbes. Usually, well, not usually, as a rule, general purpose medium is a complex medium because you want to provide that buffet selection of nutrients to microbes that you can potentially isolate. Next week, we're going to use TSA, that agar, TSA tryptic soy agar, again, for isolating microbes from the environment around you. So I suggest, think about several places that you want to sample around you. They can be, the only request is make it accessible. I mean, if you want to sample Dean's chair, it's not going to happen. Okay, but other than that, feel free. Okay, so it supports growth of different microbes, but some microbes are very, very picky. And these microbes are called fastidious. They require some sort of extra nutrient, growth factor to support the growth. Okay, so digested soy protein is not enough for them. These microbes have to be grown on the enriched media. Okay, some of them require vitamins, some of them require specific amino acids. Now, here are examples of two enriched media. Okay, the blood agar and the chocolate agar. We're going to start with the chocolate agar, which has no chocolate whatsoever. It has blood, sheep blood, that was heated up, so all red blood cells got cooked pretty much. They got destroyed. Okay. Now, so that's why it's chocolate. And actually, you can see here the colonies. It's the microbe called Neisseria. I can guarantee that you've heard of two common diseases that different species of Neisseria genus cause. One is gonorrhea. Neisseria gonorrhea causes that sexually transmitted disease. And another is meningococcal infection. Neisseria meningitidis causes meningitis. Surprise, surprise. One of the most severe forms of bacterial meningitis that we know. Okay. Now, Neisseria requires, it will not grow on the TSA successfully. You have to give it cooked blood. This, it's okay. Think about some of the national foods. This is the blood agar. In this case, blood is not cooked. Actually, sheep blood is added to the agar, so erythrocytes, red blood cells, stay intact. What you can observe on the blood agar is the process of hemolysis. Some microbes can destroy red blood cells. When these microbes grow on the blood agar, they form the clear zone around the colony. Does that make sense? Other microbes will not destroy, or they will destroy, but that destruction will be incomplete. You can see the partial hemolysis here, the green dot. Now, if I would ask you, what do you think in terms of potential to cause the disease? Microbes that don't do anything to red blood cells and microbes that destroy them, which should be more pathogenic? Yes, the ones that are hemolytic, the microbes that can destroy red blood cells, are usually more pathogenic. Does that make sense? So we can differentiate different types of microbes, and we'll chat about it. And before we go any further, think about, okay, let's take TSA, the one that you used, the agar plates, used just now, the blood agar right here, and the chocolate agar. Chocolate agar and blood agar has something in common, and TSA doesn't have it. What do they have in common? Blood. It's a different form, right? And what is that unique precious component of the blood that bacteria crave so much? Iron, exactly. There's a poem by Rudyard Kipling, Iron, called Iron is Master of them All. It's pretty much about bacteria, okay? They really want iron. When we get sick and we get fever, what happens to the iron in the blood, like free iron? Does that go up? It goes down. Huh? Yeah. We have a sequestration of iron in the blood when we get sick. So microbes, if they get in the bloodstream, will not start just to grow, because you add iron and microbes they love it. Almost all of them love it. I mean, they grow much better on something that contains iron. Neisseria must have it to grow. You see? So that shows you the example of enriched medium compared to the general purpose. When you grow microbes on the medium, you can get different types of culture. Culture can be pure, like here. Culture of E. coli, culture of Micrococcus luteus, those yellow colonies that you saw on your agar plates. Culture of Serratia, that's Serratia marchensis, that red, purple, whatever you want to call this color. Okay? So, pure culture, when you have only one species growing in your media, is most common, obviously. We're not going to get pure culture very often. Now, this is pure culture. This is Serratia only, nothing else is in there. Does that make sense? It's a pure culture. The cultures that you've got on your plates are so-called mixed. Okay? 2, 3, 4, whatever species you can isolate and grow together, it's going to be mixed culture. So every time you sample, like when you're going to go around and sample the environment, you don't really know what you're going to get on your plate. Okay? One thing, by the way, word of warning, if you think about sampling the bottom of your shoe, I strongly suggest against it. It is gross, but you won't be able to isolate any species because it's going to be like a lawn. It's like bacteria covered with fungus. Seriously, there's no isolation. I mean, find something less dirty, like urinal. I'm not kidding. Now, mixed culture is when you expect several species, like on this culture, when you see yellow micrococci and white E. coli. Now, contaminated culture is when you expect your culture to be pure, like this culture of Serratia. But it gets screwed up by some unidentified contaminant, right? That's contaminated. So you see the difference between mixed and contaminated. In contaminated, that second species is unwanted. All right? So we really have to be careful in order to avoid contamination. But to be honest, we don't do a lot of stuff with the pure culture through the course. We're going to be working mostly with the mixed cultures, but we will chat a little bit more about it. I've already introduced you to the concept that media can help you to identify microbes. The first one that I wanted to talk about is selective medium. So selective, when you say the college admission is selective, what does it mean? College admission is selective. So not everybody gets in, right? Yeah. Same goes for media. Not everything gets to grow on it. Media has something that prevents microbe or microbes of a certain type to grow on it. Does that make sense? The whole idea, right? Like you don't see Saints fans sitting in the Atlanta Falcons sector, ever. So some sort of inhibition. It can be chemicals, it can be concentration of salt, it can be pH or something else. For example, this is the Mannitol salt agar, MSA. We're going to work with that. So mannitol is a sugar, okay. S is salt. So mannitol salt agar has the concentration of sodium chloride at 7.5%. It's high. It's a lot. It's really salty. This concentration, the only microbe that can grow, well, not only, but among the clinically important ones, is Staphylococci. You already encountered one, Staphylococcus epidermidis. You've heard about Staphylococcus aureus already, right? So Staphylococcus can grow at the high salt, others cannot. So you specifically isolate a genus, okay? So that's your salt component. Now, what about mannitol? Well, it turns out that some species of Staphylococcus can ferment mannitol. Does anyone remember from, I don't know, biology, 1510, 1200, high school biology, maybe biochemistry? What is the most common product of fermentation? Not the most, but common product of fermentation. Some sort of an acid. Lactic acid, yes. So if microbe can ferment mannitol, it will produce lactic acid. What's going to happen to the pH of the medium? It's going to go down. And if you have phenol red, you see red stuff, yes? That's the indicator. When pH goes down, your colonies become yellow. Does that make sense? See the change in color. Now, let's say you have 3 species. You have Streptococcus pyogenes. Well, okay, you have strep, Staph epidermidis from your skin, and Staph aureus from your nose. You put them all on the MSA. Is strep going to grow? Based on what I told you. Strep, not staph, strep. Can it grow in high salt? No, it cannot. So we eliminate, right? There's no growth where the strep is. Staph, both species, are they going to grow? Yeah, but Staph aureus can ferment mannitol. Staph epidermidis cannot. So we can say, we have 2 Staphylococci species, but one is mannitol fermenter, one is not, so that's probably what they are. Does that make sense? So this media, first of all, it selects for the particular microbe to grow, and then it also allows you to differentiate, and we'll get to what that type of media is called. Now here are some examples of selective media. I will highlight few of them that you may run to, or they represent a good example. For example, tomato juice agar. What is the pH of tomato juice? Some of you actually mentioned that in a quiz. 4. Okay, so it's acidic. It is so pH 4 is freaking low. Not everything is going to grow at that pH. Most of the microbes won't be able to. Lactobacilli can grow this pH, and this tomato juice agar will be selective for it. What do they do, lactobacilli? Well, yeah, they can make like things like yogurt and stuff. Okay, that makes sense. So that's one thing. You see, they are adapted to grow in the acidic environment. Buttermilk is slightly acidic, maybe not so slightly. Yogurt is acidic. Those are acidic environments. So they are selected to grow in acidic environment. Another example that I want to show you here, Macconkey Agar. It contains bile. Which part of the body can we find bile in? Anyone know? Huh? Gallbladder, digestive system, right? So the purpose of this is to grow enteric microbes. Not just microbes that grow in the that live in the digestive tract, but enterics, intestine. Does that make sense? Gram-positives are not going to grow on it. So you can isolate. Saburod agar. We're going to use this next week. Saburod agar allows you to isolate fungi. It inhibits bacterial growth. Now, one thing that I want you to keep in mind, in microbiology, one of the things that confuse students sometimes, the rules, the laws, like smaller laws, are often stretchable and fuzzy. When we say Saburod agar inhibits bacterial growth, it doesn't mean that bacteria is not going to grow on it. It means that they're going to grow slow. That makes sense? So I mean, if you would compare the size of the colony, bacterial colonies on subrod after the same amount of time will be tiny, 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 tiny little, and will be full size on the regular TSA plate. Does that make sense? Inhibition doesn't mean completely prohibiting. It means very much slowing down. Right? Any sense on this? Any questions on the selective? There should be one. Do you have to memorize all this media? No, you don't. You don't have to memorize the names of the media. You don't have to memorize what they're made of. No. Because unless you work with it, you not use it, you lose it. Okay? But what you really have to understand is the concept of selective media. So if I tell you Macconkey Agar, which contains bile salts, prevents growth of gram-positive bacteria but supports growth of gram-negative microbes isolated from intestines, which would be the best description of it? I give you answers. You have to understand the description that I gave was about selective. Does that make sense? Did I make myself clear about that? That's what I want you to focus on, the concept, rather than brutal memorization of names and components. Does that make sense? I will expand on it after we discuss what the differential media is. So imagine that you have a general purpose media here that maintains growth of a number of species. They can be of different sorts, those species, but they all, for some reason, appear the same on the plate. If you use differential media, magically, well, not magically, it's a science. Magically, well, it's a science. Microbes will appear in the different colors, for example. That makes sense? So differential media contains chemicals that will give microbes different colors. Does that make sense? So you will still get 5, 6, 7 species. It's not that restrictive, right? But it will allow you to differentiate species based on their appearance. If you would look at the list, you will see mannitol salt and Macconkey. Two types of media that we just discussed is being selective. So can the same medium be selective and differential at the same time? Apparently so, yes. Okay. Does that make sense? For example, switching from MSA to Macconkey Agar. When you put your sample on Macconkey Agar, the only species that you're going to get to grow are gram-negative enterics. It can be Salmonella, it can be Enterobacter, it can be E. coli, it can be Enterococcus. Bunch of them, okay? They will grow. So that was a selective component, only gram-negative enterics. What about differential component? It contains lactose in the indicator. Some of those microbes, the enteric microbes, they can use lactose, sugar, right, as the energy source for fermentation. What is the product of fermentation? Lactic acid. Again, acid. Neutral red will change the color from red to yellow. Now my next question would be, say you have two microbes, microbe A and microbe B. They are both gram-negative. They are both coming from human intestines. A can use lactose for fermentation, lactose fermenter. B cannot. It's a tougher question. Which one do you think will be more pathogenic? One that can use lactose or the one that cannot? What do you think? Okay. You know, you can't get away with just answering. I will ask you to expand on that. Argue. Why? You're right. You're right. Why? Huh?
It's more likely to be a host for bacteria.
Say again.
More likely to be a host for bacteria.
What? Okay, so lactose increases the growth capacities of bacteria. But then you may say, well, if they kind of overgrow, they may cause disease. So it's from human intestines. And the ones that can digest lactose, can use it in fermentation, usually non-pathogenic. Why? Lactose, where does it come from? No, lactose. Yes. Do we eat it? Yeah. So, since we normally eat dairy products which contain milk sugar, lactose, microbes that can digest it are considered to be normal components of our guts. Does that make sense? Because they kind of evolved with us. They are used to the fact that host, us, eats dairy. Okay? They flourish in the same nutritional same nutrients as we do. Okay, the ones that can't, kind of suspicious. Why they avoid lactose? They probably come from some place where lactose is not abundant. Probably they're pathogenic. Does that make sense? No? No? Not really? Now, one thing that I want you to understand, if I ask you, does that make sense? I do not expect this. If it doesn't, I expect no. It does not make any sense. Can you explain it again? So if it doesn't, let me know. Now look. Okay. I'm sucker for analogies. I came up with another one. You meet someone in New Orleans, and that someone claims to be local, and that someone says, I know. Okay, you meet someone in Germany, and they say, oh, I'm German, I know. I live here all my life. You tell them, well, let's go and eat blood sausage, which is pretty much blood, cooked blood. And the person starts to vomit. Well, he's not German. If you're German, you have to eat sausage. Like you meet someone in Russia and they don't like vodka. They're not Russians, right? They don't drink vodka. Okay? If they can't tolerate it. So you know that this person is kind of from other place. Same goes for your intestinal bacteria. If they cannot digest lactose that is commonly present in your intestines, they are from some other place. They're not normal. By the way, if you refer to lactose intolerance comes from your own genetics. If you're lactose intolerant, that's because you don't have a microbe. And you weren't most of your life. When you started, when your mom fed you, you were fine. But eventually. Anyway, so that's differential medium. Example of differential medium. Again, you don't have to know the list, but you really have to know the concept. A couple more examples. Triple sugar iron agar. So it contains 3 different types of carbohydrates and iron. So this is your uninoculated one, right? This one is inoculated with a microbe that does not use any of the sugars in the process of fermentation. So it just grows there. Okay? This is definitely a fermenter. You see that yellow part? That's acid. Why there is a little bit of red up here? Obviously there's no growth up on top. Growth is only at the bottom. What's the problem with the top? Yep, oxygen. So this is anaerobe, right? This guy apparently ferments the sugar, right? But it's not afraid of air. So it's it's probably air tolerant anaerobe, the one that can survive in the presence of oxygen, or facultative anaerobe. And here you can see black. This means sulfur reduction. Okay? Again, the same stuff that we discussed. Iron sulfide is formed up top but not at the bottom. So it's probably some kind of aerobic bacteria which also can reduce sulfur. Does that make sense? So see, look, this medium gives you an idea of oxygen requirements, nutritional capacities, will or will not use certain sugar, and ability to reduce sulfur. You can diagnose a whole lot of microbes using the approach of differential medium. Probably the ultimate differential medium is so-called chromagar. I don't want you to feel that those examples, pictures that I show you, are detached from clinical practice. Although this picture is taken from already outdated textbook on microbiology, in almost every issue of the Microbe magazine, which is published by American Society of Microbiology, look at the last page, and they have ad for Agar. Agar is the product that's routinely used in the clinic. It's used to diagnose most common pathogens of the urinary tract. So you see E. coli, Klebsiella, Proteus mirabilis, Enterococci, Staphylococcus saprophyticus, and Staph aureus. They're all written there. And each name appears in the color of colonies of that organism. So colonies of E. coli are going to be like dark pink, Staph saprophyticus light pink. You see? So you can collect urine specimen, or you can collect the urethral swab, plate it, and diagnose if you have a suspected urinary tract infection. Diagnose right away. Give it a couple of days to grow. Does that make sense? You see? Before we move forward, any questions on the differential versus selective? Again, you don't have to know that freaking list, okay? But concept, I will test you on that. You can absolutely, you can expect questions. Okay. Now, this was all for bacteria. So bacteria, they can grow by themselves. They grow independently, right? What about viruses? What do they have to grow? What specific, well, host. Yes, host. Can you expand on it? Cell. They need a cell. They have to be inside of the cell, whether it's a bacterial cell, protozoan cell, or mammalian cell. So in order to cultivate viruses, which are obligate intracellular parasites, we have to grow them in a cell culture. You see, said some viruses can be grown in a cell culture. There's one that scientists spend like 20-something years trying to figure out, hepatitis C. It's a pain in the butt to grow in the cell culture. Okay. I don't think they now have the good model for it. So cells. How do you collect cells? How can you make? So cells on this picture, they grow on the surface of those flasks. You see they covered with a purple medium. You see? So they grow on the surface in so-called mono layer. So it's a layer that is one cell thick. How can you collect them? I mean, where do you get cells from? Where do you get cells from? You can take from skin. Tissue, yeah. Pretty much any tissue. You can take blood, isolate monocytes. If cells can divide by themselves, almost every time you can make the tissue, the cell culture out of it. You all know what circumcision is. Those little pieces of skin at the left of the circumcision. Ever wondered what they can be used for? They're cell culture of human foreskin fibroblasts. So those pieces of skin are used to isolate fibroblasts, cells of the connective tissue. You can buy them. I mean, not the skin pieces, obviously. You probably can buy them too. I never inquired. But cells, absolutely. There's a non-profit organization in Virginia that sells it. Okay, you can take a piece of fat, add some trypsin, the enzyme, the protease. It will break down the proteins that attach cells to each other. Like shake it, put it on the plate, wait for some time, remove all the clutter, and you're going to have some cells stuck to the surface. Okay. Now, what is the problem with the normal cells from tissue? What they do? Like, okay, you grow cells. They occupy all space. You collect them, put them in a bigger space. They occupy all space. You collect them, put them in even in a bigger space. What's going to happen to them after about 4, 5, maybe 6 generations? Yeah, they're going to die. That's why we don't die of cancer all the time. Okay? Cells die. Now, cancerous cells or transformed cells, tumor cells, don't die. How can we transform cells? What can cause mutation in a cell so it becomes cancerous? Oxidation, yes. We can treat cells with chemicals. So DNA mutates and they start to grow indefinitely, something else. More likely DNA. You have to get this into the genome. How can you mess up DNA? What do you have to do with the cells? Except for treating them with chemical. That's a good approach. Yes, you can throw in some mutant proteins. You can throw in some proteins that will mess up DNA. You can just put them in ideas. UV light. Like, melanoma is caused by overexposure. Same. You can put them under the UV light. That's kind of a lot of work. Where else can you get cancerous cells from? Yes, bingo, from somebody's tumor. Have you heard about Henrietta Lacks? It's quite an... Huh? Henrietta Lacks was an African-American woman who developed cervical cancer, like mid-50s. And she went to the hospital and she went through surgeries. She finally succumbed to the cancer eventually. But your tumor got excised before that. And the tumor tissue was used to prepare the cell line. Pretty much, it was broken down into individual cells. It was grown in the flasks like that. And those cells were called HeLa, Henrietta Lacks, H-E-L-A. These cells are responsible for probably a good third of all discoveries in molecular biology, microbiology in the world. You can find them in pretty much any lab. They're like bread. They're so common. So this woman lives in those discoveries. That's her legacy, essentially. But it turns out her relatives were not very happy with the fact that her cells are used so deliberately by scientists. What is the problem? What do you think? And they are rightfully unhappy. Yeah, they were used without her consent. Why those people who used it without her consent were not prosecuted by law? In order to give consent, you got to have an idea of consent. In 1956, there was no idea that patient has to consent to using his or her tissue for scientific purposes. It was normal to cut somebody's tumor and use it in the lab. Nobody even asked themselves. Okay? By law of 1956, it was not a problem at all. It was normal. Now it's a problem. Let me ask you why it's a problem now. Why you cannot simply take my blood and start using it? Why do you have to get my consent for scientific research? Huh? Well, it's a vague thing. What's so unethical about not asking me? What kind of personal information can you get from my cells? that's probably going to be the least of my problems, believe me. And I don't. Well, at least last time I got checked. What can you get from the cells? What type of information is stored in this? Yes, genetic information. That makes sense? Genetic information is everything about you. And which business will almost kill for that type of information? Huh? Say again? Not just medical. Medical insurance companies. Because they would know that, say, you have genetic predisposition to diabetes or high blood pressure, so they would manipulate the rates. And it's now a big concern, a growing concern, as we get into the age of genomic sequencing that would cost now. Illumina recently announced that they're going to sequence human genome for $100 in 2 years. So what's going to happen, like you have a baby, you come to physician first time, he takes sample of blood, and next day you get the full list of all genes that your baby has. That's pretty impressive, really. But insurance company sees something and says, oh, I don't like this. I'm going to jack up the price for insurance. That makes sense? Huh? Exactly. It's not even pre-existing condition, but it becomes a tricky issue. So, family of Henrietta Lacks became very became very unhappy about this whole story because the genome of those cells got sequenced. And the genetic information, not only about Henrietta Lacks, but about the whole family, was exposed to the scientific community. Well, speaking honestly, I will not go online, we will not search, we will not look into it, right? I mean, we're not going to do that. I'm not that much interested in the genome of Henrietta Lacks or her relatives, but it creates a precedent. And this is something that I want you to think about. Kind of interesting, isn't it? There's a book actually named Eternal Life of Henrietta Lacks that describes this whole thing. Cells are maintained in the medium. You can see the list of the components. No, you don't have to know them. Pretty much cells have to have nutrients and other stuff. So this medium for cells, is obviously defined, but cells themselves can be considered complex. You don't know the chemical composition of them, right? You can also grow microbes in the live organisms, like eggs, or animals, or even plants. You can see tobacco mosaic virus here compared to the normal leaf of a tobacco plant. Now, before we get to animals, it sounds kind of funny, tobacco mosaic, who cares about it? Let me tell you something. First, the names of the plant viruses are the best. I don't think anything can beat the bushy stunt tomato virus. That indeed causes tomato to be bushy and stunted in terms of the growth. Second, USDA and companies that link to agricultural business, they give a lot of money to researchers who study pathogens of the plants. I was working in the University of Nebraska and our funding got cut. We were working on human pathogens. People from plant pathogens were laughing at us. They were filthy rich. Monsanto gave them money, USDA gave them money, like all other companies gave them money. Because this is the food supply. That's not some lousy hemorrhagic fever. Okay, people can get hungry. Animals. You see influenza replicating in mice. In some cases, that's your only option. Animals. Like when you need to grow worms, you have to go to some rodent models and grow worms in the rodent models. Okay. Eggs. You see that woman that inoculates eggs with influenza virus? The vaccine that you get, the virus, inactivated virus that is in flu vaccine, is grown in eggs. So let's ask ourselves why eggs is not a perfect medium for growth.
I mean, something could be like wrong with.
With the egg.
Yeah, like they can have like a genetic mutation.
So they can be genetically altered. Good answer. They controlled, and to kind of preemptive answer, these eggs are pathogen free. So they have no viruses or bacteria. They're special. What is another disadvantage? Yeah, excellent. Yeah, people with egg allergy cannot receive the vaccine. People eating tasty animals? They would probably care less about eggs than about killing cute chimpanzees. But yes, I can relate to animal protection. Space considerations? I mean, in my fridge, eggs take up a lot of space, and we talk about 5-6 dozen. Imagine millions of them. It's a lot of space. Yeah, we eat a lot of eggs. I was never more happy when I bought like 90 in Walmart with some crazy discount. They were selling these huge boxes. They lasted for like 4 weeks. Huh? Oh, in Walmart they're used to everything, I think. What is another disadvantage? What do you think? Who produces eggs? And are they machines or living? They're living things. And living things have a tendency to die unexpectedly. But a couple of years ago, there was a huge shortage in eggs in the United States. So among the companies that make flu vaccine, it was almost a panic. They were really freaking out because they didn't have enough eggs to make flu vaccine. Well, guess what was killing chicken? Flu. The bird flu. So bird flu was killing chickens. So they didn't lay eggs, which were used to make vaccine against human flu. Isn't that ironic? Okay, so unstable. So why we kind of talked about qualities, why they were used in the first place. We didn't have any cells at the time. They're like, each egg is a giant cell, essentially. I'm going to ask you, why don't we switch from eggs to cells? Yes, but we can grow influenza virus in a cell culture like this. Okay, so it will. No, actually, cells are much easier to work with. I mean, you stick them in an incubator, they grow in the incubator, take them out, you collect cells. It's really easy. That shell, all that crap that eggs produce, it's just too much bother. Why don't we switch to?
Is it because it's more compatible?
Okay, compatibility. We can pick a lot of different out of a lot of different cell lines, so we can pick whatever we want. Pathogens. We can make cells that have no pathogens whatsoever. It has nothing to do with science. Absolutely nothing to do with science. Huh? Who? Ethics? Oh no, cells don't care. Cheaper. Cells are much cheaper than eggs. Huh? What happens when you change something in the drug, which vaccine essentially is a drug? What do you have to do when you change something in a manufacturing process? Patenting, well, it's up to you. Huh? You have to test it and you have to get approval for it. and you're going to spend like millions of dollars to get approved vaccine that is not better, that it may be 20-30% cheaper, okay, just for the sake of moving from eggs to cells. Who cares? Now there's a work on making a universal flu vaccine, so we don't have to get a new flu shot every year, okay? Huh?
So that would be wonderful.
That would be lovely. If they will get to the clinical trials, they will probably start with cells, because it's a completely new drug, so you can just do whatever you want with that in terms of the regulation. That makes sense? Okay, we got through this. Let's wrap it up, and we'll chat about incubation and isolation on Thursday. There is a great likelihood, not actually likelihood, I can promise, there will be a quiz on Thursday that will cover the studying microbes. So you see that red font right there? So if you go all the way up until cells, it's going to be multiple choice. There's going to be 15, I think 15 or even less than that questions, 10 questions. Okay? That makes sense? Think 10 multiple choice and 5, and you can use your notes. Yes.